Hello students, good morning. Today we are going to start a very interesting chapter, that is biotechnology. It comes under unit 9, chapter 11 and 12 are included in this unit. That is, first chapter is about the principles and processes of biotechnology, and the next chapter is the application of biotechnology. Today let us discuss about the principles and processes. That is chapter 11. We are going to start today. Very interesting. And then nowadays we need vaccines for immunization process in large amount, in large scale we need. And also antibiotics to kill the disease causing bacteria. We need in large scale. All these antibiotics and vaccines and enzymes are produced in large scale by using living organisms, by using prokaryotic cells. So, that technique is known as biotechnology, using living organisms. Biotechnology means it is a technology that use living organisms. That's why it is called biotechnology. What is biotechnology? It's a technique of using living organisms or enzymes, enzymes to produce products and processes useful to human beings. Even making curd, bread or wine was also considered as a form of biotechnology. But nowadays, this making bread, wine and curd are not considered as biotechnology, because nowadays they refer biotechnology to processes which are genetically modified organisms, which use genetically modified organisms to achieve the same on a large scale. Nowadays they use genetically not only the prokaryotic cells, but they modified the gene of the prokaryotic cells and they use for producing antibiotics and vaccines. That processes only are now considered as biotechnology. Even in making curd, bread or wine, microbes are used. They play an important role in curd, lactobacillus and then yeast. These are all living organisms. With the help of living organisms, we get all these products. So in previous days, these processes were also considered as biotechnology, but nowadays they alter the gene of the microbes and they use for producing products and any processes that are useful to humans. They refer that as biotechnology. Other processes that are included under biotechnology are in vitro fertilization. It also comes under biotechnology, and nowadays they synthesize a new gene and then they use it. That is also a biotechnological process. And then developing a DNA vaccine and then correcting a defective gene. If any individual is born with a defective gene, it leads to many disorders, genetic disorders. Nowadays they correct the defective gene, and that also comes under biotechnology. Now, the European Federation of Biotechnology has given a definition of biotechnology that combines both the traditional and modern views. What is the definition given by EFB? European Federation Federation of Biotechnology. The integration of natural science and organisms, cells, parts of the cells or molecules for products and services. This is the definition given by EFB, European Federation of Biotechnology. Now, let us study in detail the principles of biotechnology. Among many, the two core techniques that enabled birth of modern biotechnology are genetic engineering and maintenance of sterile ambience. genetic engineering. Already I explained you, nowadays they use the genetically modified organisms in biotechnological process. So we have to modify the gene of the organisms, and afterward we can use for producing products. Now, so techniques to alter the chemistry of genetic material, that is genetic engineering. Techniques to alter the chemistry of genetic material. DNA and RNA are the genetic materials. They change the chemistry of this genetic material, the makeup of the genetic RNA and DNA, and they introduce this into the host organisms. And when they introduce this into the host organisms, this change the phenotype of the host organism, because when they introduce this genetic material into the host, then what will happen? The genotype will be changed, and then if genes are changed, then the phenotype also will be changed. And this is one core technique. And next, while growing the desirable genes, sometimes the desirable microbes, sometimes the undesirable microbes also will be grown along with this. So we have to maintain sterile environment. That environment will allow only the desired microbes to grow in large scale, but it will not allow the undesirable microbes to grow. So maintenance of sterile ambience in chemical engineering processes to enable growth of only desired microbes or eukaryotic cells in large quantities for the manufacture of biotechnological products like antibiotics, vaccines, enzymes, and etc. These two are the core techniques in biotechnology. And we know and we appreciate also the sexual reproduction, it permits variation. We appreciate the advantages of sexual reproduction over asexual reproduction, because this sexual reproduction, it provides opportunities for variations and formulations of unique combinations of genetic setups. Gametes are fused together, so variations occur in sexual reproduction. And then in asexual reproduction, the genetic information of an individual is preserved. And then in traditional hybridization, it includes the inclusion and multiplication of undesirable genes along with the desirable ones. In traditional hybridization, when we multiply the desirable genes, along with that undesirable genes also will be multiplied. But we need only the desirable genes. But this drawback is overcome in genetic engineering. This include creation of recombinant DNA or new DNA, and use of gene cloning and gene transfer. These are all different techniques. And isolate and introduce only one or a set of desirable genes without introducing undesirable genes into the target organism. Here in genetic engineering, only the desirable genes are identified and then selected, and they are introduced into the host organism, and they are allowed to multiply inside the target or host organisms. So it is more advantageous than the traditional hybridization process. And then now we studied that on DNA will be, our genes will be introduced into the host organism, and they allow the host organism to multiply. Along with the host organism, the desirable gene or DNA will also get multiplied. And then DNA, what is DNA? It's a part of it is present in the chromosomes. And what is a gene? A part of the DNA. A part of the DNA responsible for the expression of a definite character is gene. So the piece of DNA, desirable piece of DNA, that is gene. Now we will study about the fate. What will happen to the fate of a piece of DNA inside the host organism. Host organism is known as alien because it is alien to that DNA. DNA is from another organism, a piece of DNA, and then we introduce it into the another organism. We take a piece of organism and we introduce it into the another one, and we allow this one to multiply in number. And now DNA can multiply only when it gets integrated into the genome of the recipient. First, this DNA, we want to multiply a desirable piece of DNA. This will be introduced into the host one, and then we allow the host to multiply. Along with the host, this desirable DNA also will get multiplied. For multiplication, this desirable DNA should be integrated into the genome of the host, genome of the recipient. Then only it can multiply. In a chromosome, there is one place that is origin of replication. Only in this origin of replication, the initiation of replication process starts. The replication means duplication of DNA. Replication of DNA or chromosome means duplication. During cell division, the amount of DNA is doubled by this replication process. Replica means copy. More copies of DNA will be produced, and that is replication of DNA. This replication, it starts only at a definite place in the DNA. That place is known as origin of replication. And then this alien DNA is linked with the origin of replication. So the desirable DNA is linked with the origin of replication of the host, so that this alien DNA can replicate and multiply itself in the host organism. And this process is called cloning, or making multiple identical copies of any template DNA. Now, let us now focus on the first instance of the construction of an artificial recombinant DNA molecule. How the first artificial recombinant DNA molecule was constructed. What is recombinant DNA molecule? First, we select the desirable DNA from any organism, and then a piece of DNA we select, and then we introduce or ligate or combine this piece of DNA with the DNA of the host, and that DNA is called, now this DNA has the original and also the new alien, original and then new alien one, and then that DNA is called recombinant DNA. And then we introduce this recombinant DNA into the host organism, another organism, and then this host organism will get this recombinant DNA. First, we introduce this recombinant DNA into the host organism, and this host organism will be allowed to multiply, and we get the desirable DNA also in large scale. It also gets multiplied along with the host organism. And then we cannot introduce this alien DNA into the host DNA. For that, we need one vector. We will study this one shortly. Now, first, how they constructed the new recombinant DNA. This recombinant DNA means what? One DNA, and it has one alien part, that is desirable DNA is also linked with it. This gene encoding antibiotic resistance with native plasmid of Salmonella typhimurium. What they did, the construction of the first recombinant DNA emerged from the possibility of linking a gene encoding antibiotic resistance with a plasmid. They they ligate, they combine the desirable gene with the what was that desirable gene? That was a gene encoding antibiotic resistance, which gives antibiotic resistance to the organism. That gene was ligated with the plasmid, with the native plasmid of Salmonella typhimurium. Salmonella typhimurium is a prokaryote. While studying prokaryotic cells in 11th standard, I told you what is a plasmid. It is a plasmid. You will study in detail about this plasmid in your 12th standard, I told you. So now this plasmid, what is plasmid? These prokaryotic cells, they have this genetic material, and then one circular autonomously replicating extra chromosomal DNA is also present in the prokaryotic cells, and that is known as plasmid. What is plasmid? It can multiply by itself. That is autonomously replicating circular extra chromosomal DNA. It is not along with that is normal chromosome, extra chromosomal, but inside the cell. It is circular in shape. And it can replicate by itself. That is why plasmid is autonomously replicating. It can replicate by itself, and it is circular, and it is extra chromosomal DNA. It's not present along with the chromosome extra. And Salmonella typhimurium is a prokaryotic cells. It has this plasmid. So they encode this antibiotic resistance. The gene that encodes antibiotic resistance is ligated, is fused with the plasmid of Salmonella typhimurium. The two scientists, Stanley Cohen and Herbert Boyer, they accomplished this in the year 1972 by isolating the antibiotic resistance gene by cutting out a piece of DNA from plasmid. First, they cut a piece of DNA. They cut the piece of DNA, and then afterwards they ligate it with the vector DNA, and after that they introduce into the host DNA and the piece of DNA from the plasmid. First, the cutting is done by restriction enzyme. Long DNA will be there, and it will be cut into so many pieces. How is it possible? By using enzymes. That enzymes are known as restriction enzymes. These restriction enzymes are also known as molecular scissors because they cut the DNA molecules. And then this cut piece of DNA was then linked with the plasmid DNA. They ligate the cut DNA with the plasmid DNA. This plasmid DNA act as vectors. They act as vectors. In malarial parasite, what happens? This Plasmodium is a malaria causing parasite. It is transferred from one individual to the another individual through female Anopheles mosquito. This female Anopheles mosquito is known as vector. So in the same way, here also one DNA will be introduced into the another DNA. One piece of DNA will be introduced into another using a vector. And also we have to link that cut DNA to the host DNA and then another DNA, and that function is done by some another enzyme, that is DNA ligase, and DNA is cut by using restriction enzyme, so they are called molecular scissors. And then this linking is done by DNA ligase. Now new combination of circular autonomously replicating DNA is created in vitro. It is created in vitro and is known as rDNA. It is known as recombinant DNA. And then this rDNA is introduced into the host. This rDNA now replicates. When it is introduced into the host, it replicates using new, that is host DNA polymerase enzyme. So for taking multiple copies, this enzyme DNA polymerase is used to make multiple copies. So 3 enzymes. For cutting the DNA, we use restriction enzymes. For linking the DNA with the plasmid, we use DNA ligase. And for taking multiple copies, DNA polymerase enzyme is used. The ability to multiply copies of antibiotic resistance gene in E. coli, this is the host, was called cloning of antibiotic resistance gene in E. coli. Cloning, that word cloning means getting multiple copies that in the host. So now you see here, look at your page, your book page number 197. Here, this is the diagrammatic representation of recombinant DNA technology. First, we take the foreign DNA and then we cut a piece of foreign DNA, this desirable DNA, and then vector. Using this vector, circular plasmid, we introduce this alien DNA into the host, it's E. coli. First, we cut this foreign DNA with restriction enzyme, molecular scissors. We mix this DNA and the enzyme, then reaction will take place and then the DNA will be cut into many pieces. And then we use the same restriction enzyme for cutting this plasmid DNA of the plasmid DNA. And then now you see same DNA we use, same restriction enzyme we use here also and here also. That is very important. We will study later on. And then this piece will be next afterward ligated in with the plasmid. Now we get a new plasmid, and this is known as new DNA, and this is known as rDNA. That is because it has one alien piece and its own piece. This for ligating this alien DNA with the plasmid, we use ligases, DNA ligase enzyme. So ligases join foreign DNA to plasmid. And then after that, this is known as rDNA, recombinant DNA. And then this recombinant DNA is transferred into the host, that is E. coli. And then E. coli, it can, when it multiplies along with E. coli, this desirable DNA also gets multiplied, and then we get more and more amount of this desirable DNA. This is the basic principle. In this way, this rDNA is synthesized. And then that process is known as cloning. And then there are 3 steps in genetically modifying an organism. First, we have to identify the DNA with desirable genes. And then we have to introduce this identified DNA into the host, into the E. coli. And then after